Bismillah Rahman Rahim. Assalamu alaikum. This is Dr. Hooria Aamir. I welcome all my students of first year and first year MBBS, BDS to this session of Cardiovascular Physiology. As dear all, you know that we have started the topic of heart physiology, and we have just touched the topic, and gradually we are going to build this subject. So I hope all of you must be following the lectures as well as the Untitled Chapter 14. So what we are going to study in this is about the functional anatomy of the heart. We can learn the functioning of any organ not until and unless we know about the anatomy of that particular organ. Then how the working is going on is going to be studied over there in physiology. What are the different events which are taking place during the course of contraction as well as relaxation of heart. We are going to study that in detail in cardiac muscle. Then we'll be studying about the cardiac output controls and the blood pressure. Now the functional components of the cardiovascular system, as you all know from the previous lecture, what are they? They are the first one, that is the conducting system. And what is that? That is the blood vessels, including arteries, arterioles, capillaries, venules, veins. Then we have a pump, you all know, that is the heart. Then the circulating fluid medium, and that is the blood. Now what are the different functional benefits of this system? The functional credits, the functional benefits to be taken out of this cardiovascular system function, they are that they help in transportation, they help in regulation, they help in protection, they help in production and synthesis. So what are these things? The transportation, so everything that is transported to the cells, either they are given to the cells or they have to be taken back from the cells, that is via blood. Now regulation of the cardiovascular system, that is both the intrinsic control of heart as well as the extrinsic control. Then protection against the blood loss. We all know that whenever there is blood loss, there is process of vasoconstriction going on, and there is you can say that the narrowing of blood vessels is going to decrease the blood loss, and there is production and synthesis of some of the chemicals also takes place. So the cardiovascular system function, to create the pump, we have to examine the functional anatomy. What is this dear heart? What is its composition? The composition is, we all know, the answer is the cardiac muscles. Then the shape, we all know about the shape, and there is presence of 4 chambers, we all know, and these 4 chambers or 4 compartments, they are being segregated or separated from one another by means of valves. Then there is presence of intrinsic conduction system. This intrinsic conduction system is responsible for the movement of action potential from SA node to whole of the body of the heart. So we are going to study them in detail, one by one. Now let's see, you can see that how the muscles of the heart, they look like, the cardiac muscle. You can see that there is presence of striations. They are short, but they are branched. They are actually branched to one another by means of intercalated discs. They are joined. They are uninucleated. They have T tubules larger and overseed discs. They have intercalated discs and they have very high magnitude of mitochondria. So we have to keep the structure of cardiac muscle in our minds when we are actually learning about the functioning of the heart. So the interior of the heart, that is actually having the 4 chambers, the 4 compartments. The 2 upper ones, which are the smaller ones, they are the 2 atria, whereas the 2 larger ones, they are the 2 ventricles. And then there is presence of 2 systems, and what are they? They are the pulmonary system and the systemic system. As we know that the heart is actually a dual structure, so there is a right heart and there is a left heart. One is dealing with deoxygenated blood and the other one is dealing with oxygenated blood. Now when we are doing this, now we know that there is compartmentalization of the heart interior into 4, and this is actually the work which is being done by the presence of the valves. Now why these valves they are there and what is their basic function? Now the function is actually to maintain the unidirectional flow of the blood, that is to prevent the backflow of the blood. Now we have the atrioventricular valves, we have the semilunar valves. Now atrioventricular valves, what are their functions? Their function is to prevent the backflow to the atria. Then prolapse is prevented by the chordae tendineae and the tension by the papillary muscles. Semilunar valves, they prevent the backflow into the ventricles. Now the functional anatomy of the heart, there is presence of the intrinsic conduction system. Now this system is consisting of the pacemaker cells, which are the 1% of the entire population of the heart muscles, and conduction pathways. They coordinate the contraction of the atria and ventricles. Now you can see, and you all should be drawing this, the conducting system of the heart in your logbooks, that there is presence of SA node, then there is internodal pathways, and then we have AV node at the junction of the atria and ventricle. Then there is a right and left atrioventricular bundle. From them there is origin of bundle branches, and then there is a ramification upwards, which are known as the Purkinje fibers. So this is actually the basic of the conducting system of the heart. If you are being asked in exams and you have what is the natural pacemaker, what is the normal pacemaker of the heart, that is the SA node. God forbid if this SA node is not working properly, then this activity is to be taken up by the AV node. If that is not functioning, in case of any heart attack, stroke or etc., then that function is to be taken up by some other portion of this conducting system of the heart. So we should be knowing that the physiological pacemaker is one and only SA node, and that is the sinoatrial node. Now the myocardial physiology actually revolves about the 2 types of cells, and what are they? They are the autorhythmic cells, the pacemaker cells, and the 99% of the contractile element. And what is that? That is the cardiac muscles. Now let's talk about the autorhythmic cells first of all, the pacemaker cells. Now what are the characteristics and how do they differ? The thing is that normally they are smaller than the contractile cardiac cells. They don't contain many myofibrils. There is no organized sarcomere structure. So that's why they do not contribute to the contractile force of the heart. So we all have to keep this thing in mind, that although they are there in the heart, but they are just active in generation of the action potential in the heart, but they are not contractile themselves. So the autorhythmic cells, the pacemaker cells, the characteristics. Now the characteristics which are very important to learn about, and yesterday you have done this, that what is the difference in the action potential over there. You can see that the resting membrane potential over here is minus 60 millivolts. And there is lot of leaky channels there, and they gradually drifting towards positive side, and when they lead to like coming across minus 40 millivolts, they form a pacemaker potential. Then we have the myogenic. The upward drift allows the membrane to reach threshold potential, which is minus 40 millivolts by itself, and this is due to slow leakage of potassium out and faster leakage sodium in, causing the slow depolarization, and it occurs by IF funny channels that open at negative membrane potentials and start closing as membrane approaches threshold potential. Calcium channels opening as membrane approaches threshold. At threshold, additional calcium ion channels open, causing more rapid depolarization. These deactivate shortly after, and slow potassium channels open as membrane depolarizes, causing an efflux of potassium and a repolarization of membrane. You can see, and all of you should know how to draw this graph of action potential, the pacemaker action potential, which is gradually, you can see, becoming less negative until it reaches the threshold voltage. And what is that? That is the minus 40 millivolts, and triggering an action potential. Then in the B graph, you can see that there is ion movements during an action and pacemaker potential, how it is going on. This is actually the story involving the calcium ion and potassium ion. Then there is state of various ion channels. You can see this is how this rhythm in the heart that is being produced, and how these pacemaker cells they function, and altering the activity of pacemaker cells. Now the pacemaker cells, they are not actually their working is not constant. I mean that their working can be altered. This is by the effect of autonomic nervous system. Now when we are talking about the altering activity of pacemaker cells, we should be knowing about the sympathetic activity. Now the norepinephrine and epinephrine increase the funny channel activity.